Okay, very good. So I think we can get started. There will be another couple of people coming along, hopefully. So extremely happy to have Duncan Robertson here. Duncan, I remember, as I'm sure a number of us do, from lecturing him. He was the guy sitting there with the special laptop that he created himself, making these amazing notes. And I got to know Duncan during the course of his undergraduate, spoke about many different topics. He then left us to go to Stellenbosch to do an MSc in Bioinformatics, and is now at the University of Oxford in the second year of his PhD. So we are lucky and very happy to have him here today to give the second of the Shocklab seminars for the year. So yeah, over to you. Thank you very much.
Thanks, Prof. Shock. So hi, everyone. I will be talking today about the research area I'm interested in at the moment at Oxford. And it's all about the graph theory of evolution. So trying to understand how we can figure out the ancestry of a species or a particular population just based on their DNA. And so please, if you have any questions, feel free to interrupt me as I go along.
Just quickly, I think if you click on more at the top right, I think you can hide all of the...
Oh, okay.
Yeah.
It should disappear as well. Yeah. Oh, there we go. Okay. Great. So I'll be talking today about a type of mathematical object called an ancestral recombination graph. But to start off with, I wanted to talk about the most fundamental process in biology, which is DNA replication. So this is the most basic element of ancestry. One DNA molecule gets turned into two. So this is this incredible molecular machine which unzips the DNA coming in through from the left, and it's then sequences, it sort of fills in the other side of the helix on either side. So on the bottom, you can see the leading strand being duplicated, and that whole complicated machine is busy replicating the lagging strand. So it has to sequence, well, it has to add on bases in reverse and then let it loose, and then add on more bases in that loop and then let it loose. And these machines are currently doing this in millions and millions of your cells right now. And this is the fundamental process that has carried us through from primordial organisms like 1 billion years ago to where we are today. So because a DNA molecule gets turned into two, the most fundamental way we can describe ancestry at a DNA level is with a binary tree. So suppose we have a genome which is just 4 letters, AAGT. If that is replicated, then we suddenly have 2 genomes, which in this case have exactly the same sequence. And suppose the second genome here gets replicated again, we now have another 2, but maybe there's a mutation. So sometimes that replication machine makes a mistake. And in this case, it's replaced a G with a C. And that means that we have a different sequence in 0 than we do at 1. And so here you can see that everything has the same, all the genomes have the same sequence except this particular node 0. And the crucial thing here is that the nodes represent genomes and the edges between the nodes represent a transfer of genetic material. And we could also write this as a matrix. Oh, sorry, before I say that, this is also happening over time, right? So in genetics, we tend to do things a bit backwards. So we base the time on the present. So the sample nodes, which are the ones at the bottom, are at time 0. And then we consider time increasing, so time going backwards as you go up. And you can think about it in terms of like phylogeny, like the tree of life. You think about things as 1 million years ago or 2 million years ago. That's just how we date things. So we can also write at least the genetic data of these nodes as a matrix. And this is a very fundamental thing in biology. It's where the rows are genomes and the columns are positions along the genome. And you can read off the sequence based on that. But a thing to note here is that we have to store this mutation, so the C to the G, sorry, the G to the C, in this column. And we've had to write out the G 4 times. Whereas if we look at the tree, all we need to do is to store the topology of the tree And we just need to know the genome of one of the nodes. And then we can navigate through that tree and read off the genomes of all the other nodes. So this tree actually compresses the genetic data because we only have to store this mutation once. And that's something that becomes more important as we go to larger scales. So now this is an example of a bit more of a complicated tree, but is exactly the same principle. So all the numbers are nodes, and the edges represent transfer of DNA between those nodes. And here, this cross, an A, is a mutation that is at site A. So I just wanted to introduce a bit of terminology. So the nodes at the bottom, the leaf nodes, are called sample nodes. And the nodes that are in the interior of the tree are called internal nodes. And an edge is just something that connects the nodes, and the descendant or child of the node is at the sort of bottom, and the ancestor or parent is what it inherits it from. It inherits the DNA from. So this all feels very abstract, but there's some really interesting trees that you can uncover in biology. Sorry, that's just the root node. And this is one of the best examples. This is the tree of our human mitochondrial DNA. So the thing about mitochondria is that they're passed in the egg cell, so from mother to child, and there's no mechanism for like mixing up of the DNA, so no recombination or anything. And it makes it incredibly powerful as a tool to understand human origins, because it means that you can trace our ancestry from mother to child all the way back to the common ancestor of all humans on the planet today. And this tree is from a fantastic paper on mitochondria, which summarizes that. So the red parts of the tree are the oldest groups of people. So in Africa, so we have some of the, because of the human origin in Africa, you can find the oldest lineages in the continent. And the map is colored by the different groups of sort of mitochondrial DNA, or what are called haplotypes, so like particular mutations that make up a kind of tapestry. So you can see that the next oldest group is currently based in India, and the most recent groups are based in sort of Russia and North America, South America. And what's neat here is that as before, oh, sorry, yeah.
Do you know about what sort of phenotypic expressions are unique to those groups, or in this mitochondrial DNA? Is that what the group for now?
So the variations are just a sort of few subtle mutations in different parts of the mitochondria, and quite a lot of them are what we call neutral, which means that they don't actually have any effect on the person. Generally, any mutation that has a seriously bad effect is going to be sort of ironed out of the population. So the variations that we use to group people by their mitochondrial DNA aren't going to necessarily give them any health benefits or deficits. Basically, almost all genetic variation has no effect. That's the weird thing. So we can use it to understand the history of a species or of a population, but it doesn't actually affect how their biology. And one thing to note here is that in this little branching here, that is the common ancestor of all humans. And this is what a particular woman known as mitochondrial Eve, that is estimated to have lived at about 170,000 years ago, give or take 50,000 years. And the method that we use to estimate that is based on the tree itself. We can figure out, based on how fast mutations happen, and given the sequences that we have from present humans, we can date ancestral nodes and then figure out these common ancestors and when they existed. So it's a very powerful tool. But unfortunately, for the rest of human DNA, this picture is not so simple. So most of our genome is what we call diploid. And what that means is that we actually have two genomes, and each of those genomes has completely different ancestry and a different set of mutations. And what I've shown here is a bit of a karyotype, which is just a picture of our chromosomes when they're bundled up. And each of the chromosomes has two what are called homologs. each of which we inherited from one of our parents. So we inherit the entire genome from our mother and an entire genome from our father. And those genomes are, in terms of the sort of population genetics, in terms of their ancestry, are completely separate entities. And that causes a lot of problems for us. So we record what we call diploid, which just means we have two genomes, as opposed to haploid, which is a single genome. And as I said, each of them have distinct ancestry mutations. And when a sperm or egg cell is formed, our two complete genomes get sort of muddled together. So quite often no recombination happens, and we just chuck, I'll say, our paternal chromosome 1, we just chuck it in there, and our maternal chromosome 2, we just chuck in there. But sometimes recombination happens where our paternal and maternal DNA gets jumbled up, and then we chuck it in. And that causes a lot of problems because it means that we no longer have a single tree that defines human ancestry in terms of our chromosomal DNA. We have two genomes, and it just makes everything horribly messy, basically. And this also means that there's a difference between an individual's ancestry and our ancestry at a DNA level, so our genome's ancestry. So you might recall with the trees, I said that if we know the sequence of one of the nodes of the tree, we can infer the sequences of all the other nodes. But if we just know a pedigree, which is just an individual family tree, if we know, say, if you know the complete genome of my father, you can't just from that infer what my genome is because you don't know which chromosomes I ended up inheriting from them. So nevertheless, these pedigrees, which are individual ancestry diagrams, are still extremely useful in genetics, but their primary use is in actually tracking traits instead of tracking DNA itself. Just as a fun example, this is one of the most famous pedigrees, which is the British royal family. And the red is indicating the presence of hemophilia, which is associated with vampirism. And it's an X-linked disease. So it occurs on the X chromosome, and it's recessive, which means that only men will, so you have to have two copies of it in order for it to be expressed in a woman, whereas you only need one copy in a man. And as a result, it's extremely rare to occur in women. But so the square in a pedigree indicates male, and the circle indicates female. And you can see poor Leopold here, and Fred, and Voldemort, whoever they are, they all inherited the trait. So these pedigrees we use a lot in genetics, but we use it to track traits over generations primarily. But we can still link it with DNA. So let's suppose we have a horrible family with inbreeding, where a mother and child had a son and daughter who then had a son and daughter. So I apologize for the example, but it makes it much easier to draw. Okay, so let's suppose we just focus on a particular chromosome. So the colors here just indicate two different copies of a chromosome, and F0 and F1 are the two homologs of the father, and M0 and M1 are the two homologs of the mother. So when the gametes are formed, one of them, so the father will choose one or two of, or either one of their chromosomes and pass it on, and the same for the mother. So let's suppose F1 goes to the daughter, M0 goes to the daughter from the mother, and F1 also goes to the son, and recombination occurs in the mother when she creates the egg. So now we have a mix between dark blue and light blue. And then let's suppose in the next generation, the daughter inherits the red chromosome from either one of their parents, and the son inherits the blue chromosome from the daughter, and they just inherit that recombined chromosome from the son. So we can now actually look at this purely at a DNA level and forget about the individuals entirely. So what we can do is just write out the individual genomes of each person in a grid like this. So those are the two father chromosomes, the two mother chromosomes, and so on. And then we can read off this diagram what the ancestral relationships are. So the genome of the father went down to the daughter, and we can annotate it with an interval, which is just the length over the genome that was inherited. So let's suppose this genome is just 10 base pairs long. What this is saying is that the entire genome from 0 to 10 was passed on from the father to the daughter. And from here on, if I don't indicate an interval, it means the entire genome was passed on. So that same genome went down to the son, and the mother went down to the daughter. And now with the recombination, the first half of M0 went to the son, so 0 to 5. And the second half of M1 went to the son, which is 5 to 10. And the intervals add up to the total genome of the son, which makes sense. If it didn't, they would have a horribly shortened chromosome and wouldn't be happy. And we can do the same with the next generation. So the red chromosome went from the daughter to the granddaughter, and so on. And in this case, the recombined chromosome went straight from the son to the grandson. So what this structure is, you might be able to see is a graph because it has nodes and it has edges. And this is actually what we call an ancestral recombination graph. And we call it that because it can actually capture recombination, because we can have multiple edges going into a single node. And it's sort of ancestral because it's tracking our ancestry through time. But the weird thing about it is that we individuals no longer are sort of first-class citizens. We put gray boxes around nodes and say that's an individual. But the only thing that the individual's effect is that recombination can obviously only occur within an individual, but we have to label them after the fact. If it's a species that's haploid, then we don't need to worry about that because each node is also an individual. So let's just generalize a bit. So an ancestral recombination graph is a directed acyclic graph, which means directed, so it comes from, all DNA comes from an ancestor that gets passed down, so it has very much a direction to it. It goes from older time to more recent time. And it's acyclic because you can't have a loop where DNA gets passed from father to son and then back from son to father. DNA can't go backwards like that through time. And it has the following properties. So the nodes, let's say it's a set of integers n, represent haploid genomes, so it's just single genomes of a fixed length, and they typically just have a time associated with them. So in the case of humans, the time will probably be the date of birth of the individual. And the edges are annotated with a set of intervals that are the regions over which the DNA is passed from the ancestor to the descendant. So in the previous example, the interval from M0 to S1 in the top right here was 0 to 5. And so we just have a graph, a directed acyclic graph, where edges are annotated with sets of intervals. And this is this fundamental structure. It's not, if you think about it, all that complicated in the way we've defined it, but it's an incredibly powerful idea because it captures much of the evolutionary biology of a species, for example. So it's a very powerful object. And the one thing to note about it is that it accounts for recombination, and it can also account for point mutations, because we can just, and like we did with the trees earlier, we can say that a mutation happened between the time that the parent existed and between the time the child existed. So yeah, I'll get to it later, but the limitation of that is that it can't handle what are called structural variants, which are bigger chunks of DNA that get deleted or duplicated. And that's just a limitation of it. So got to open this demo. But what this, yep.
So it can deal with single nucleotide variation. Can it deal with multiple single nucleotide variations?
Yeah, you can have as many single point mutations as you like, but they can be in direct sequence, but they're treated as independent things.
So can one treat a structural change as being just multiple single nucleotide variations?
No, because it's almost impossible for a bunch of single mutations to happen in sequence at the same time. The probability of that is practically zero. So, and it's also not how they get passed along. So that structural variants get passed along generally as a chunk. They're not like independent little mutations. It's one entity. So you could kind of pretend and hack things like that, but it would be very dubious. I'll probably go a bit long if I show this demo, but all I wanted to show is that this is another example of an ARG, and you can see when it splits. So this is a genome that's then was replicated into two lineages that go out in either direction. And you can see recombination when you have two edges that are going into a node. So this node here that I'm circling and that node there were within one individual, and they got recombined to form that node. And these edges will be annotated with intervals that describe which bits of DNA were passed along. So this object, it's not very nice to work with because it's not a tree, and trees have a lot of benefits, both computationally and intuitively. We kind of have a decent understanding of a tree, but not a graph with lots of edges like this. And it turns out there's a very useful thing we can do with an ARG. So the thing you have to realize is that while we can't represent our complete genome's ancestry with respect to everyone else as a tree, we could look along the genome and choose a particular position. And so that particular position, let's say the 5th position in chromosome 1, that is a single nucleotide that we inherited from a single parent. It's impossible to have a single nucleotide that comes from two individuals. So if you take any position along any genome, that position will have been inherited from a parent, and that parent would have inherited it from another parent. And no matter how far back you go, you'll always only have a single parent. And that means that you can't have any sort of loop in the sort of structure you form. So if you scan along a genome, you can find a tree, an ancestral tree for each position. And it turns out that if we find the sequence of all the trees that go for the full extent of our genome, we can form something that's actually equivalent to an ARG, and it's something that we call a tree sequence. And it's a very useful data structure because trees are much easier to work with, and we can do a lot of algorithms much more quickly using them, and they're also easier to visualize. So ARGs have been around as a concept since the 70s, but they are incredibly difficult to simulate and also to sort of try and uncover from real data. And tree sequences are an innovation that came up quite recently that my supervisor at Oxford, Jerome Kelleher, kind of pioneered. And they enabled quite a revolution in the field because tree sequences can be simulated much more quickly than the sort of ARGs that we were talking about before. So I wanted to show some of the, let me first actually explain how we can get a tree sequence from our example. So here I've just squished up that ARG that we showed before. And in this case, we have a single recombination event that occurred halfway through the genome. So if we want to turn this into a tree sequence, we just need two trees. And bear in mind here that this we're treating on the left as one tree, just because there will be a common ancestor that I just haven't pictured because I don't have the space. But if you look at this graph, if we just follow it, G0 has a parent that's D0, that's has a parent F1. So you can find that in the tree there. And similarly, G1 goes to S0, goes to F1. G1 goes to S0, goes to F1. And the key thing is with the recombination. So between 0 and 5, the parent of S1 is M0, and we can see that here. And then between 5 and 10, the parent of S1 is M1, and we can see that here. So you can see from this that these two structures are telling us the exact same information. And it just so happens that the one on the bottom is for most purposes better to work with. So I wanted to show some, and that this position where the recombination occurred is called a breakpoint. So I firstly wanted to talk about how we can use this to simulate data from scratch. So if we want to just generate a bunch of genomes, there are two main ways we can do it. So the first way is called backwards in time. And with that, we choose a number of individuals, and we choose a genome size. And we then simulate backwards in time, finding common ancestors for different genomes. So if I take this group of people, while we all, I'm pretty sure, have different parents who are not closely related to each other, I guarantee you that if we were to go far back enough in time, we would find a single common ancestor, at least for each sort of chunk of our genome. And so these algorithms just do that. It's based on something called a coalescent model, which is it's a Poisson process where you're going backwards in time, and the events that can happen are either a recombination happens or what we call a coalescence, which is where two lineages become one. And you just go far back enough in time until you get a common ancestor. And my supervisor's group created this tool called MS Prime, which is incredibly efficient because it uses tree sequences, and it can simulate enormous genomes. Like, I can simulate a genome of 1 million base pairs long with a population of 1,000 people, and I can do that in a minute on my laptop. No previous tool could come anywhere close to that. So it's a very powerful tool. The downside of them is that it's very hard to model things like selection. You have to make a lot of assumptions in order for the maths to work, for the simulation to make sense. Whereas on the other hand, the forwards in time simulators are a lot more flexible, and they're also perhaps more intuitive. So that's where you start with a collection of individuals, and you simulate their reproduction forwards in time. So you create new offspring, you cut off the previous generation, and then you simulate mutations as you go along. You simulate recombination, and you just sort of progress the population through time. But the problem with it is that if you do that without simplifying the structures you create, it grows exponentially. Because if you go back to human history, most of the lineages that existed have gone extinct. Like, most family lines end at some point. If you store all of those extinct lineages, it just becomes impossible to handle computationally. So the hard thing with these algorithms is to constantly simplify the trees that you're creating as you go forwards in time. And SLIM is one of the most commonly used tools for that, which is also based on tree sequences. But it's incredibly powerful in that it can model lots of very complicated evolutionary dynamics. It can even do spatial ecology type things. It can model populations in space with dispersion between, like, continents and things like that. So these forwards in time simulators have a lot more flexibility, but they're also generally slower. So there's a trade-off between them. But both of these are pretty invaluable for the whole of population genetics, because we use these to try and understand how evolution works. We use it to try and understand the history of populations, how, say, like a lot of our understanding of human origins stems from a lot of these simulations. Yep.
Can you use the forwards in time one to generate synthetic data that you can then train backwards in time algorithms on?
You could, but there isn't really a sense of training anything, because these aren't AI algorithms. So you could use a forwards in time simulator to create tree sequences, and you could actually take those and kind of simulate them further back in time using a backwards in time simulator if you wanted to. But neither of these algorithms are trained in any sense. And so the weird thing about it is that in population genetics, we often don't really care that much about the sequence or how realistic the sequence is biologically. We care about the structure of the ancestry, so which groups emerged when and how they're related to each other.
But presumably with the backwards in time, there are parameters that you need to set?
Yeah, so the main ones are just recombination rate and mutation rates. And those are things that we fit from real data for a given species. But besides that, there are ways to make it a bit more biologically accurate in the way the sequence is determined. But as I said, usually that's not really that important for population genetics, at least with how we use these simulations. Right, so that's simulation, but the thing I wanted to talk about in a bit more depth is going the other way. So suppose we have real genetic data from a species. Can we infer ARGs? So can we infer the ancestry given those sequences? And that is a much, much harder problem. So, oh, sorry, before I get to that, I just wanted to quickly work through how MS Prime works. So the neat thing about these simulators is that you first simulate just the ancestry, not caring about mutations. So in this case, we've chosen a population of 3 individuals. So, and they're diploid, so there's 6 nodes. And the algorithm then goes backwards in time and keeps going until we have a common ancestor across the whole genome. And in this case, the simulation just did 1 recombination. So we have 1 breakpoint at the 686. And these trees tell you how each genome is related to each other genome. So the neat thing is that we can assume generally that mutations are neutral, so they don't change the fitness of an individual. And that means we can actually throw them on after we've simulated the trees. So there's another function called sim mutations that just takes a mutation rate and then throws on mutations on the branches. And all it looks for is to make sure that the longer the branch is, the more likely it is to have a mutation. Basically, the longer a lineage is, so the more time that passes, the more likely it is that a mutation will occur in that time. And at the bottom here, these arrows just indicate where the mutations occurred. So this is the general workflow that we do, where we simulate the trees, so the structure of the trees, and then we throw on mutations afterwards. And that has worked surprisingly well.
Presumably, so the structure of the tree, if you run the simulation multiple times, it could give completely different structures?
Absolutely. So it's very much a random process. So you'd have to set a seed in order to prevent it from giving a different result each time. And the same with the mutations. So I wanted to talk about just how efficient it is to store genetic data in this tree form. So remember that with this tree sequence, if I know the genome of a single node, I can infer the genomes of all the other nodes just from this structure. So all I need to do is store each mutation once, and I need to store the nodes and how they relate to each other in the trees. And I just need 1 sequence, and that's all I need to store all this data. And it turns out that that is incredibly efficient. So what this plot is showing is how much storage space you'd need to store the genome, a simulated genome of every human on the planet. So 10 billion complete human genomes. So the pink line, the solid line, is what we'd need to, if we're storing the data in a traditional matrix, where the rows are individuals and the columns are variants. And you can see that as we go up in genome size, we end up needing 25 petabytes to store 10 billion human genomes. Whereas if we stored it in a tree sequence, which is the solid blue line, then we would only need a micro SD card to store the genome of every human on the planet. It would fit into a terabyte micro SD. So that is a compression algorithm that's saving 5 orders of magnitude of space. And what's more, it's not actually losing information, it's gaining information. So it's not only lossless, but it's also storing the ancestry of the sequences in addition to the sequences themselves. So that's why these tree sequences are so powerful in modern genetics, is that we now are dealing with enormous genetic data sets, and our computational methods are starting to really struggle to handle that volume of data. So by storing data in a tree sequence, not only do we save an enormous amount of space, but we also understand more about the data. Yep.
Is it clear why from here 10 to a million, the rate of increase is different from a million to 10 billion?
Yes, so it's because the algorithm, so the tree sequence inference algorithm it scales at order of so it's n log n plus n. So as you go to larger genome sizes, the n term dominates. So it becomes a linear scale. It starts to scale linearly. whereas with smaller genome sizes, it scales logarithmically. Yeah.
Really the expected rate of mutation also plays a role in its asymptotics expectation of how this function will evolve. Because I mean, if I imagine and say I'm growing a cell line and I am mutating them by applying step by step a different chemical mutagen, physical mutagen every single time, they have no chance of affecting the resistance to mutagens. That would require the kind of changes that would occur would just be so rapid as to, I don't think you can scale things more efficiently than the matrix.
No, actually. So the thing is that mutations are actually very cheap to store. They require very little space because you're only storing each one once. And
enormous changes in one thing, so that you mutate the exact same pair as well as another pair, you change.
Yeah. So in that scenario, it would probably you'd be right that it would be difficult in practice to use tree sequences because you need to have some sort of variation in common between the individuals. So if there's too much mutation, it can be hard to figure out what the trees are. So in practice, it probably wouldn't be a good use case for it, you're right. But in terms of storage, it's not actually that much of an issue, but in practice, you wouldn't. So generally, when we're using tree sequences, we're looking at populations that occur on an mutations that occur on an evolutionary scale, which are actually very rare. Hence why we all share almost all of our DNA with each other. So this, we're talking here about the size we need to store simulated genomes, but a lot of what we want to do is with real genomes that we've sequenced. So there are numerous benefits to being able to infer these tree sequences from data. First is that we can understand population history and ancestry from the structure of the trees. And secondly, as I said, it's a lossless compression algorithm that I should say at least 2 orders of magnitude of compression. So it saves an enormous amount of space. So the challenge is that to infer infer the ancestry from DNA is a very, very hard problem that's only recently being properly solved. Yeah, I'll talk about that a bit later. So all of the tools that we use today to infer ARGs came out quite recently. So my supervisor, Jerome Kelleher's one came out in 2019, and it was the first one to use tree sequences. And it's also currently the only tool that can handle massive whole genome sequencing datasets. Then these later tools, which were also developed in other groups at Oxford, have different specialities. So Relate is quite a lot more accurate for like recent past. So it's used for human ancestry studies. And this tool called ARG-Needle is used for what are called genome-wide association studies, which is basically a regression where the predictors are mutations and the response are phenotypes. So you're trying to predict, given these mutations, will this person be taller? Will this person get cancer? And if you infer ARGs and then do this regression, you can save an enormous amount of computation time. And this is a very new line of research, but it's proving to be very promising. So I wanted to show some particular examples of how we have used TS-infer in particular. The first one is in trying to get an ARG that encompasses recent human ancestry. So this is work that was done by a past PhD student of my supervisor, Wilder Wones. And so Jerome Kelleher and Yan Wong, they're the supervisors of my PhD. And what they did here was to try and understand the ancestry of modern humans, and particularly try to understand how we relate to our recent relatives, so the Denisovans and the Neanderthals. And the neat thing is that we actually have ancient DNA from some of these sort of recent relatives of ours. So what they did was to infer ARGs from the biggest whole genome sequencing datasets that were available at the time, which were 1,000 genomes and the Simon's Genome Diversity Project. So from that, they built up trees, which represented the ancestry of all those individuals. And then they could thread in these ancient DNA samples from Neanderthals, Denisovans, and a group of people called the Afanasivo in Russia, who existed around 5,000 years ago. And that from that, they could incorporate them into the ancestry and figure out how we were related and when our populations diverged. So they then had a tree sequence for our combined human ancestry. And what they then did was actually associate ancestors with particular parts of the world based on how closely related they were to more recent samples. So they ended up with this amazing plot. So what this is basically a tree that's been distributed across a map to show the passage of time, to show the passage of a population through the world. So the purple represents our oldest ancestors, and the orange is our more recent ancestors. And you can see that the oldest lineages were in Africa in general. Obviously, you can see it's not a very precise algorithm, especially. And as you go up into Eurasia, the ancestors become younger and younger. So this was directly created from the tree sequence, adding in additional location data in a non-parametric model. I don't quite know the full details of it. Yeah, so if you recall from the mitochondrial DNA, some of the oldest the oldest lineages besides Africans are in India and southern Russia. But the thing is, this algorithm that they did to spatially cluster it was very imprecise. So you shouldn't read too much into it in that sense. But it gives you a rough idea of how the sort of DNA passed along across the world. And the other example I wanted to show is about SARS-CoV-2. So it turns out coronaviruses are actually capable of recombination, but obviously in a very different way to how our cells do it, because they have a single genome and it's RNA, not DNA. But that actually made it very difficult to track the sort of evolutionary development of the virus over time, because all the methods that virologists tend to use are based on phylogenetics, which is the field that's concerned with ancestry on a species level, on like a longer time scale. And phylogenetics has a tough time dealing with recombination. So the existing methods weren't handling that very well. They had tricks to get around it, but what my group did, led by Xin Zhan, who's a postdoc in our group, was to infer an ARG from the largest SARS-CoV-2 dataset, which is about 2 million genomes. And they could store the full sequence of 2 million genomes in 58 megabytes. It's pretty neat. And they can also very rapidly update it as more and more data becomes available. And it's provided a lot of insight into the ancestry of the virus and how the different strains developed that previously wasn't possible to figure out. And I just have a neat diagram from the paper here. So on the right is a phylogenetic tree of the COVID virus that shows the different lineages. So here's the alpha variant, there's the delta variant. And on the left is actually a particular tree in the tree sequence that was inferred from the data. And this shows just how closely they match up. So the tree sequence has captured the same patterns that the other methods did. But in addition, it's containing a lot more data about the recombination between different lineages. And recombination was actually an important factor that in virus developing into more aggressive forms. And what we're currently working on is inferring ARGs on the biggest data sets currently available for human genetics. So the first one is 100,000 Genomes Project, which is run by Genomics England, which is the provider of sequencing technology for the NHS. And it currently has 80,000 individual whole genome sequencing results, which is literally petabytes of data. And the other one is the UK Biobank, which is 200,000 individuals with whole genomes and an additional, I think, 300,000 with lower quality sequencing. And the challenge of inferring ARGs from these data sets is enormous because, firstly, just a single chromosome in the UK Biobank is 16 terabytes of data. It's a single 16 terabyte matrix. And even though the TS-Infer algorithm is incredibly efficient, it's still an enormous challenge. But we've actually, so Ben Jeffrey, he's a computational scientist in our group, has actually figured out the optimizations to make it work and get it to fit into memory and everything. So we can now, we've done chromosome 20, which is one of the smaller ones, but we can do a complete chromosome if we have maybe like 100 to 200 nodes on a cluster in about a week or two. So it's pretty remarkably fast considering. And the challenge that I've been working on is quality control. So the problem is we end up with these tree sequences, which literally have millions of trees, 150,000 samples, millions of ancestors, and we don't really have many tools to actually figure out what's going on, like whether those trees make sense, whether we've made any mistakes, because this has never before been possible to even get sort of infer ancestry on this kind of scale. So we're developing a web app that will help us visualize these trees and figure out problems with the inference algorithm. And it's working pretty well. So I've kind of given an overview of how we're using ARGs in genetic research, both in terms of simulations and in terms of inference. And now I wanted to get onto what I'm actually going to be doing in my PhD. And that is to address the biggest problem with ARGs, which are the assumptions that all the genomes are exactly the same length and only single nucleotide variants can exist. So that immediately means that you can't model structural variants, and this is just a visualization of them. So deletions, insertions, duplications, inversions, where the sequence flips around, and translocation, where a big chunk gets moved from one place to another. So with long-read sequencing, this new sequencing technology, we can now identify structural variants much more easily in real data. And that's led to an explosion in medical research where we've uncovered a lot more about human genetics regarding these structural variants. But population genetics haven't caught up with those advances yet because we don't really have any tools to simulate structural variants. And the other problem is that we can't simulate multiple species in an ARG because multiple species almost always have different genome sizes. For example, the lungfish is a very close relative of humans, but it has a genome that's 20 times larger than ours. Sorry, it's not 20, 14 times larger. So we can't capture evolution on a larger scale using ARGs. That's where phylogenetics comes in. So if we can generalize to handle varying genome lengths, we can encompass the full scope of evolutionary biology. We could capture the entire history of entire history of life in a single graph, in theory. And we have an idea for how to do this, which my supervisor, Yan Wong, came up with a few months ago, actually, which is called a genetic inheritance graph. And it's actually quite a relatively simple generalization of an ARG. The idea is that instead of annotating edges with intervals, you annotate edges with coordinate transform functions. So they're functions that tell you if you're at this position in the parent, that position corresponds to this position in the child. And that allows you to say if there's a duplication, this position in the parents maps to two different positions in the child. Or if it's a deletion, you just have to shift it in coordinates. So as an example, here I have a genome where the arrows indicate chunks of DNA. And here I've supposed that the green chunk got duplicated, the blue chunk got inverted. And this is a parent and this is a child. So we need to find a function that maps this genome on the left to that genome on the right. And we'll also find a function that does the converse. And if we can construct a graph with these functions linking up nodes, in theory, we could capture the full extent of human genetic variation. But in practice, it's going to be very, very hard to get it to work properly. And just to do things like to extract trees from this graph is not a trivial matter anymore, because we have to track coordinates as they change, coordinate systems as they change over all these structural changes. So I'm still in early days of it. It's going to be to get it to the point that we can simulate DNA. It should, well, that's the goal of my PhD. So I'm hoping to get that done in 3 years. But to get it to the point that we can infer these structures from real data is probably going to be at least 10 years. But if we can succeed, it will create a new field of genetics. So it's quite an exciting project, but very, very challenging.
You started trying to investigate like the basis of such functions. Like obviously there's the whole whole gene duplication thing.
Yep.
Like, have you been looking at the underlying biology of like what causes these things to happen? So that would inspire you to list all these possible some things.
So we at the moment don't really care about exactly the biology of how these sort of variations occur. What we're concerned about is just how we can model all of them. So the list isn't very long. Like this is the full list of possible structural variants here. They all emerge from lots of interesting different mechanisms. So generally duplications and deletions happen when recombination goes wrong, and inversions also can happen with recombination. So the biology of them is absolutely fascinating, but at this stage, we're just trying to get a simulator that can throw deletions and duplications onto genomes going forward in time. And we don't really care if the duplications are a realistic length or if they occur in the right places. As I mentioned, in population genetics, we don't care that much about the sequence itself. We care about the ancestry of sequences through time and the processes that occur over longer time scales.
I was kind of asking, how sure are you that they're all of them?
Oh, pretty sure. It's literally the only real possibilities in terms of like how you can alter a sequence given the machinery of how DNA is duplicated.
There's not very strong Turing complete sort of.
No. No, not at all. Yeah, so that was all I wanted to present. I just wanted to thank my supervisors, Jerome Kelleher and Jan Wong at the Big Data Institute. This picture's taken on one of the 2 sunny days of the year. And my colleagues in the group, Savita, Ben, Xing, Kertian, and Daiki, and the convener of my PhD, Julian Knight, and my funders, which are the Clarendon Fund Christchurch and the Medical Sciences Doctoral Training Center. Thank you.
Thank you very much for a fascinating talk. Are there any more questions?
So when you're defining the R where you have the intervals, do you actually use integer indices, or do you, because of these things where the length of our genomes change, do you look at like sub sequences, like highly conserved, between this genetic, like say between this gene A and between gene B, we have some sort of random-ish noise which doesn't have any phenotypic differences, but we will see it evolving. Do you like look at those like finite sequences and actually index them with integers? Like, do you look at that?
It's a good question. So traditionally ARGs are treated as having continuous genomes, which makes no biological sense because the sequence is a discrete sequence of nucleotides. But it makes mathematical sense because in order to simulate ARGs, you generally assume that a mutation never occurs in the same place twice. And they did it as a convenience by if you sample a continuous variable, it's almost impossible. Well, it's very unlikely that you'll end up with 2 of the same numbers in a row. So historically, we've done continuous genomes, but with the gig that we're developing, we've decided to use a discrete genomes as coordinates instead, because it makes a lot more biological sense, and we don't really need to use that sort of trick anymore. But in terms of like labeling subsequences and things like that, generally we wouldn't, because again, it might sound counterintuitive, but we're not actually that concerned about genes in particular. So we're not trying to annotate regions of a sequence with particular functions. We're treating it as a bit more of an abstract thing, which is just genetic variation over time. And we're trying to understand the forces that alter genetic variation and trying to model things like sort of evolution and selection. But in order to do that, we don't actually have to make the sequences realistic or have particular functions. It's just, it would be nice. It's beyond our understanding at the moment.
How do you define a linear meaning if translocations can occur? Like if I've got a particular, let's say I have two people's genomes and one of them has a little bit of a wild mutation, and I'm looking between 0 and 5. Like, how do I know if something hasn't been inserted there?
Yeah, that's a great question. That's probably one of the biggest challenges we're going to face with how. So the reason that nobody has tried to do this before is that as soon as you make genome size variable, you have a problem with matching coordinates between different genomes, and that makes it incredibly tricky. So
future directions, right?
Yeah, but there is a trick that's being developed, which are called graph genomes, which is a bit confusing because it's also a graph built from DNA, but it's a graph that's only built on a bunch of sequences that we have. So it doesn't concern itself with ancestry whatsoever. And it's basically based on a bunch of clever computer science of trying to match subsequences in different sequences together. So try and say like this bit is duplicated, and we can map it to this bit in another gene. And so you can use this graph structure to relate genomes of different size to each other and figure out how one coordinate maps to a coordinate in another genome. So there are solutions that have been developed for it, but they're incredibly tricky to actually implement.
I wanted to ask a couple of questions. Can you include epigenetic information in this?
As far as I know, it hasn't been done. It's changes to your genome that occur in your lifetime. So there are a bunch of cellular processes that can add labels, these sort of methylation groups and a bunch of other things. They can add them to the DNA in your genome, and then the cell reacts to those groups in different ways. So your body can repress a certain part of your genome in certain cells, or it can activate them in certain cells. And say if you have a very stressful childhood, these changes can accumulate and mean that you have a shorter lifespan.
Does it not matter when you measure the genome of the person?
The thing is that those epigenetic changes don't alter the sequence. They're chemical additions to the molecules themselves. So they're basically like an additional sequence on top of your genome. So your genome itself will change randomly in different cells just from random drift, but overall it stays pretty much the same. The epigenetic changes are much harder to measure, but these amazing long-read sequencing technologies can actually sequence methylation groups. But the main thing is that, again, with population genetics, we're primarily concerned with very long timescales. And so epigenetics is irrelevant because it occurs within one lifetime. And I think even doing things at the level of like the past few generations is very rarely done in the field. So yeah, I haven't heard of it being done, but I'm
that chunk of things. The epigenetic changes certainly carried on for a couple of generations afterwards.
Yeah, and I've heard there's some interesting evidence about that. But unfortunately, we have way too little data to really figure out. I think it's pretty unlikely to carry on more than a few generations to our knowledge. And unfortunately, it's a very frustrating field because we have basically no data. Like we're talking about those trees going back millions of years or even thousands of years, but all our genome data is either in the present day or it's some horrible ancient DNA samples from a few thousand years ago. That's it. So in population genetics, we probably have some of the most frustrating data in the world.
Yeah, and that's why I was asking about creating synthetic data.
Yeah.
That you can then test your methods on.
Yeah.
To see if it's giving something realistic.
So that's done quite extensively so with the inference algorithms, we test them by simulating data and then inferring from the data. And then we can check, do we get back, so we can simulate it with a particular model. So like a demography model of X populations moving out of Africa. And then we can infer ARGs from the sequence data that we generate and check, can we reproduce that demography model? And that's how we kind of validate everything. So simulations are fundamental to population genetics because it's how we validate almost everything we do. Again, because we have no good data to work with, so we have to make it up.
And the other question I had to you talking about the web app. Are you using any topological data analysis?
No, not at this stage. So what we're primarily doing is visualizing mutations across the genome, across time, so we can map where mutations are occurring in evolutionary time and where they are along the genome. And that's very helpful for detecting whether there's problems in our inference algorithms which bunch up mutations in weird ways. And we also do it to visualize edges in the graph, see how long the edges are. So basically chunks of DNA that between recombination breakpoints. And so we're doing quite, and then we visualize a lot of these population genetic summaries like metrics and things. But there has been some work doing topological data analysis in the field, but it wasn't particularly fruitful because it takes too long. But it would be something that could be fun to explore. It's just quite hard when the data is that massive.
Let's see if there are any other questions here. Any other questions from anybody here? If not, thank you again for a fantastic talk.
Hey, I had a question.
Oh, yes, that's all.
Oh, hi. Is this Yan Wang the same Yan Wang who collaborated with Richard Dawkins?
Yes, it is. So he's an evolutionary biologist. He wrote a book with Richard Dawkins on evolution, and he worked as a BBC science presenter doing crazy stunts. Like they made a boat out of ice and tried to sail from England to Ireland, and it failed, and they had to abort the mission. And he also almost got bitten by a bunch of rattlesnakes at some point. He's a very interesting person.
That's wild. So
Yeah.
I have a strange personal connection to him, which is when I was about 13, I emailed him a computer program. Well, I actually emailed Richard Dawkins a computer program, which was like an evolutionary simulation program. And obviously Richard Dawkins didn't reply, but he replied, and he like gave me some pointers and
That's
encouraging. And the weird thing, I don't have the email because it was like many email servers ago, but I do remember his name. It was Yan Wang. So
wow,
it must be the same guy.
what a coincidence! Sure.
Small
Yeah, he's a really
world.
lovely person. He's very much a golden retriever in personality. He will respond to everything. Yeah, he's great.
So Tally, you should maybe email him again and see if he's still got the original messaging computer program.
Yeah, he might.
Yeah, that's wild.
Yeah,
Sorry, that's not directly related to your
no,
talk,
no,
but
that's a funny coincidence.
really interesting.